Hello, everybody. My name is Berge Minassian, speaking from the University of Texas Southwestern. What I tried to put on my first slide is a picture that summarizes most of child neurology. You'd be surprised, but let me explain. Basically, in child neurology, we have a patient on the one side, and then in the middle, we have the big black box of the brain. The brain is the most complex structure in the known universe, and one day we will figure it out. And we know a lot about it already, but it's still quite black and opaque, and we don't really know how it works. And that's the organ that we're trying to address in our patients. But on the other side, we know the causative defect in the gene for most of our patients, for the vast majority of our patients. So many of our patients have single gene defect disorders now. The mutation in the missing gene manifests in many ways. For example, if a gene product is needed to sustain the brain, if you lose that protein, then you have a neurodegenerative disease. Another example is if you need a particular protein in a very specific part of the brain, and if that protein is not there, you can have a focal abnormality, like a focal epilepsy that may require surgery. But the most common situation is if it has to do with developmental diseases. So for most of our patients, the gene is required during the development of the child. And as you know, the brain uses the vast majority of the genes in the genome. Therefore, it uses many thousands of genes. And unsurprisingly, there are several thousand different genetic causes of neurodevelopmental problems. In each case, basically what is happening is we are allowing the brain to develop while missing a critical protein. Now imagine the future. The future is where we can replace this protein and allow the brain to develop normally. How far is this future? This future is not far at all. So imagine the new neurology, the new child neurology of the future. We're talking about all these phenotypes that we know that we see in the clinic, where we are basically unable to help much, and we give sad news to families. We will have a whole new set of thousands of phenotypes of children where the problem has been solved at the root, allowing much better, if not normal, development. So even today, we are able to deliver the missing gene brain-wide in newborn animals. The animal studies, whether mice or larger animals such as monkeys, are clearly showing that if we treat an immature newborn brain, we can distribute the virally delivered gene product very widely across the brain. Now, if we do gene therapy in older children or in adults, we cannot. We're currently limited at about 20% distribution in the brain. So 20% of the cells will get the gene product. So there's a huge advantage in child neurology compared to adult neurology, for example, and especially in our diseases that start so early because they are developmental, that we could potentially fix the problem brain-wide early on in the newborn child. The issue we have is because there are these thousands of diseases, how do we know what disease is going to befall a particular child before it's too late? That requires newborn screening for thousands of diseases. How can we do that? Well, $100 genomes are almost here. Currently, a genome sequencing costs as little as $600. That's already affordable, but it's heading towards $100 soon. This means that any child who is born, we can do a whole genome sequencing and identify in case they have a missing gene that will cause grave damage to their development, and try to replace that gene as soon as possible and allow the brain to develop normally. What about adult neurology? How is the situation different? Indeed, there are adult neurological conditions that are single gene defect disorders. Huntington's disease is one, several forms of ALS, some forms of Parkinson's disease. But by and large, adult neurology, at least the genetic forms, which are very, very common, are caused by polygenic disorders where multiple genes have variants that contribute to the phenotype. Let's just take the most common, by far the most common example, and that's Alzheimer's disease. And this slide just shows susceptibility genes for Alzheimer's disease, and you see there are so many. So adult neurology is similar to child neurology in many, many cases. The only difference is that it is mostly polygenic and therefore more complex. But if we learn how to replace a missing gene in child neurology, maybe we can do the same in adult neurology. We just would have to break a bigger set. So in this slide, I try to show that. Let's say we needed 5 different gene variants to cause Alzheimer's disease. If we only have 3, you do not get Alzheimer's disease. So replacing the defective genes in two of the genes would be able to fix the problem. So what we will be learning in child neurology with gene therapy will be applicable to adult neurology, which affects all of us and everybody that is related to us. Even in adult neurology, we can think of treating with single gene interventions. Here is an example again from Alzheimer's disease, and although Alzheimer's disease, the causation is complex, but it is thought today that the major final common pathway is the formation of these misfolded tau proteins, which aggregate, accumulate like this, and lead to Alzheimer's disease. What if we could replace one of these tau proteins with one that no longer is able to receive the next tau protein, and thus prevent the aggregation, and thus prevent the progression of Alzheimer's disease? So monogenic approaches to treatment of complex diseases. Since we spoke about neurodegenerative diseases, let's go back to pediatrics and look at the childhood neurodegenerative diseases. For some reason, in childhood, neurodegenerative diseases frequently present with epilepsy, and they are called progressive myoclonus epilepsies. This is an incomplete list. I filled as much of the page that I could. There are several others. I want to just say that most of these progressive myoclonus epilepsies are lysosomal diseases, and I highlighted those in yellow. And that has a particular significance, which we'll talk about as we go forward in the talk. So what is the vector that we use to replace a missing gene? It is a virus called adeno-associated virus. If you look at the top right of the picture, you see the great big adenovirus particles, and the arrowhead is pointing to an adeno-associated virus, the AAV virus that we use. This is a tiny little virus that was discovered as a contaminant of adenovirus preparations. This virus does not replicate, does not cause disease, and it has lived in humans as long as humans have existed. It is able to cross the blood-brain barrier. It is like the bacteria on your skin or in your guts. It lives with us and knows us, and we disregard it normally. What we do is we remove the gene contents of this virus, shown in the picture there in red and blue, and we replace that with the missing gene that we want to replace into the patient. It can accommodate most genes, but not all genes. It has a size limit, and there are certain genes that are too big for us to be able to do gene therapy. But for most genes, it can fit, and it will deliver the gene. The virus can be delivered IV because it's able to cross the blood-brain barrier, but the amount that it crosses the blood-brain barrier with is relatively small. And so IV approaches require a very large amount of virus, and then you end up treating all the other organs while your target is the brain, and sometimes it's not so good to treat all these other organs. So we prefer an approach directed to the brain itself. You can inject into the brain matter itself, or as we prefer, we inject in the CSF space, any place in the CSF, but lumbar puncture is one approach, and the virus will distribute to the brain. Here is an early experiment by Dr. Gray, my colleague who leads our preclinical gene therapy group here. And as you see, the virus will distribute broadly. This virus is producing a protein called GFAP, and you see, GFP, and you see how it's widely distributed in the brain of the mouse. But again, this extent of very wide, near complete distribution occurs in neonatal mice, and it is not nearly as good when you use older mice. So if you're dealing with a disease where the pathology is restricted to a very small area, here is an example. In this particular disease, it's the enzyme that contributes to making dopamine. These patients have this condition which results in them looking like baby Parkinson's patients, and the problem is in the subcortical parts of the brain. And so if you target the substantia nigra in this case, or the basal ganglia, the virus will distribute in the entire nucleus because it's such a small area. And already in 2012, we had great successes. As you see, this frozen child, and the virus is injected and the gene delivered, the motor phenotype improves, and the condition is essentially. So bottom line, with small foci to treat, it's easy, it's doable, it's all good. Even in older children, you don't need neonates. As I mentioned, in neonates, we get this type of widespread distribution across the brain. But for older children, we don't get distribution that is sufficiently widespread, and we get basically partial treatments. The example I showed you previously, because it's such a small area of the brain, we get complete distribution. What I'm showing you here is the special case of lysosomal disorders. Lysosomal disorders benefit from a phenomenon called cross-correction. Let me explain. So a lysosomal soluble enzyme, as it is being made, acquires the mannose-6-phosphate addition to the protein. And this mannose-6-phosphate modification is what mediates the transfer of the protein into the lysosome. Now, lysosomes, through the vesicular systems of the cell, fuse with the cell membrane, and the lysosomal enzymes can float freely in the interstitial space. Now, the cells have a way of recapturing these so that they're not wasted, and so there's a mannose-6-phosphate receptor at the cell surface. And so the protein swims in the interstitial fluid, and then is captured by other cells and internalized and transported back to lysosomes. So what happens in this situation is, if you treat 20% of the brain cells, let's say in an older child with a lysosomal disorder, those cells become factories to distribute protein to the other cells. And gradually all the cells acquire enough protein, and the problem is really fundamentally corrected across the brain. So this is the phenomenon of cross-correction, which is a particularity of lysosomal proteins and lysosomal diseases. Some of you may know how we treat a form of lysosomal disease called CLN2 Batten's disease by injections of protein into the CSF space. Brineura is the name of the drug. The reason that protein works is because, again, by cross-correction, it distributes from cell to cell. The problem with that treatment, of course, is you have to inject the protein on a regular basis, whether it's once a month or whatever the frequency is. Whereas in gene therapy, you give the treatment one time, and then the cells themselves distribute to the other cells through the phenomenon of cross-correction. That's why I highlighted all these lysosomal neurodegenerative diseases in children, and all of these benefit from this cross-correction, and the results are looking very exciting for all these diseases with gene therapy. And I'll show you an example later. But I'm going to switch to Lafora disease, which is in orange here. And this is the disease that I've been working on for a very long time. We discovered the genes for this disease and are trying to understand it fully, but we haven't fully understood it yet. But we want to treat our patients in the meantime, as explained before. Before I go into that, let's go back to this question of the older children. Like I said before, we're looking into the future where we can easily do gene therapy on newborn children. But meanwhile, to prepare those drugs and be ready for this new future in child neurology, we need to do the experiments and treat older children. And as I said, in older children where the entire brain is affected, we have a ceiling of about 20%, and we can only treat about 20% of their cells. What can we do to improve that as we advance towards the future where we treat all these kids early on before they have so much damage? Here are just 4 things I listed. There are other things we can do. Number 1 is we can engineer proteins in such a way that the protein, even if it is not a lysosomal protein, will behave like a lysosomal protein and will exit cells and enter neighboring cells and give the advantage of cross-correction. So we and others are working on methods to change proteins that are cytosolic, for example, to behave like lysosomal proteins and cross-correct. Another question is the immune system. You might ask, why don't we simply redose the patient? If the first dose gives us 20%, why don't we dose again and again till we get to 100%? The reason is because we give a very large amount of virus in gene therapy, 1 × 10⁻15, a quadrillion virus particles. And although the immune system doesn't mind this virus normally, when it sees such a huge amount of virus, it will react. No problem happens with the first injection, but if you try a second injection, you get 0 additional distribution because the immune system blocks it. So can we mediate the immune system to allow us to have multiple administrations, redosing? And indeed, there's lots of work going on in this area, and there's quite great advances that should lead us there soon. Third, can we evolve a better AAV viruses that distribute more widely than the current virus that we have? And indeed, much work in this area going on, incidentally, including by groups related to the company Moderna that made the COVID-19 vaccine. That's just an aside, where viruses are being developed that distribute better, evade the immune system, etc. And finally, Can we open the blood-brain barrier? As I said, the virus does pass the blood-brain barrier, but can we open the blood-brain barrier transiently and safely to allow much more access to the virus? And that, I'm just going to show you an example of that. So here we open the blood-brain barrier using focused ultrasound. So you see on the right side where the black arrow is, the hippocampus blood-brain barrier has been opened using focused ultrasound. On the left, the red arrow is intact. And we do the gene therapy during the brief period where the blood-brain barrier is open. And you see in the colored picture B in the middle, the large amount of brown indicating huge distribution of the virus and protein production, GFP, in the opened hippocampus, but not the unopened blood-brain barrier. So this is a simple approach that we are using to allow much wider distribution. Let's return to Lafora disease that I mentioned before, a non-lysosomal neurodegenerative condition. Just briefly, this disease starts around age 14, 15, and until that time, the child is perfectly healthy. As you see here, in this young girl looks perfect at age 14, and then she starts having myoclonic jerks, visual hallucinations, and a gradual dementing condition, as well as increasing intractability of her epilepsy. And finally, she becomes in a constant status myoclonicus state, eventually into vegetative state. The whole condition lasts 10 years or more, and it's a horrible condition. The myoclonus becomes completely constant, and there's nothing we can do to help these kids. It's an autosomal recessive disease. Meanwhile, the family may have had other children who see what's going on and know that they're at risk for the same. This is Chelsea when she was 21, and here just a brief video to show you what they're like over time, and then she died sometime after the state. In the brains of these children are these massive Lafora bodies that you see in these various pictures. And these Lafora bodies consist of an abnormal form of glycogen. On the left side, you have normal glycogen. On the right side, you have the glycogen that is in Lafora disease. And the difference, as you see, is that the chains of glucans are longer in the Lafora glycogen, and this makes them precipitate, accumulate into Lafora bodies that drive the disease. Glycogen is synthesized by the enzymes listed on the left side, and Lafora and Malin are two new enzymes. They're the causative defects of Lafora disease. And again, part of the black box. We know a lot about them at this point, but we don't know everything about them, and we don't exactly understand how they are important in preventing the normal glycogen that you see on the left become like the Lafora glycogen on the right. Meanwhile, our patients are dying and we want to help them. And as I said, we can do that with gene therapy, and we are doing gene therapy in Lafora disease, but we can also intervene at other places, like at the RNA and the protein. And our target does not need to be the causative gene. So in this case, I'm going to give you an example of where we're targeting a different gene than the causative one, namely glycogen synthase. Glycogen synthase in green here is the enzyme that elongates the glucan chains. So whatever the mechanism of formation of these abnormal glycogen in Lafora disease, in the end, it's glycogen synthase and only glycogen synthase that can add glucoses together and make these long chains. So what if we downregulate glycogen synthase? Can we help Lafora disease? And that's what we did here. We used antisense oligonucleotide against the RNA of glycogen synthase, and we injected that in mice to downregulate glycogen synthase and see what happens. And indeed, the abnormal glycogen accumulation normalizes, as you see here. The Lafora bodies improve dramatically, as you see in the top right graph. And if you look at the color pictures, you see in the bottom right corner is the animals treated with the antisense oligo against glycogen synthase, and the Lafora bodies cannot form when your glycogen synthase is slowed down. And the inflammation in the brain shown with the brown GFAP signal here goes away when you treat these mice with the antisense oligo against the glycogen synthase. So you get rid of the, you reduce glycogen synthase, you get rid of Lafora bodies, you get rid of inflammation and neurodegeneration in the brain. This treatment is waiting to start clinical trial soon. On the top left is Lafora himself. In the rest of the pictures are patients with Lafora disease, most of whom are deceased now, but there are many that come all the time, and we're hopeful that we can start our clinical trial soon with our antisense oligonucleotide to treat these patients. And imagine if we could treat them early on, you know, when they're born and prevent the whole thing, if we only knew early on that they had the diagnosis, if we could only do newborn screening on them, which we now can with the decreasing costs of genome sequencing. I will now show you an example in dogs of Krabbe disease. This is, unlike Lafora, which is not a lysosomal disorder, Krabbe is a lysosomal disorder. And these Krabbe dogs are treated with gene therapy, gene replacement for the missing enzyme. And I want you to see what happens to these puppies with Lafora disease. With Krabbe disease. This dog has Krabbe disease and is quite sick, as you see. And then the team here, Dr. Bradbury, Charles White, and Steve Gray, they give the gene therapy, replace the missing gene, and this is what these dogs will look like after gene therapy. So dramatic improvement because of the phenomenon of cross-correction. Now, here's an example of CLN7. This is another lysosomal disease. This CLN7, the protein product, is not a soluble lysosomal enzyme, so it will not benefit from cross-correction. It's a membrane-bound protein. This little boy's name is Jojo. He was diagnosed with this. His family moved to Dallas from Arizona so that they can support our gene therapy efforts. They raised several $1 million for us to have a program in gene therapy for this rare disease. They knew all the while that by the time we get everything organized and start in patients, by the time we finish all the preclinical work and get to the clinic, the boy may be too advanced. And indeed, he was too advanced by that time. He died around the same time that we actually started the clinical trial. He and his family raised the funds, but he did not benefit from it. But the other children who were diagnosed later, were younger, did benefit. I'm showing very preliminary video here. This is a small study of 4 patients, and this kid from Paraguay came and benefited from the gene therapy that Jojo and his family fundraised for, and she had the CLN7 gene release. This is extremely preliminary, but we already see improvements. Even though the protein product in question is not a soluble lysosomal enzyme and does not benefit from cross-correction. Slide again with all the kids we're losing all the time and all the kids that are suffering all the time. But now we can diagnose them early and hopefully make them whole again and create the new pediatric neurology of the near future. Thank you for your attention.